Hello, everyone, and welcome to our weekly update. This is Sabrina Paganoni. I'm calling from the Healy Center for ALS, the Massachusetts General Hospital. And I'm here today with our patient engagement team, Katrin Small and Alison Bullat. So thank you so much for joining. And as always, we're giving people a few seconds to join from the waiting room. And I see that people are still joining. And also I have to say we've seen really an increase in the number of people watching the recordings. So really thank you so much to all of you who are joining live and asking questions live, and then also to the people who are watching the recording. And I would say that, your questions really help um, everyone, and also help the people who are watching the recordings, because then they get more out of it. So thank you so much for participating and for asking great questions. If we can go to the next slide. I'll just present again where we are. I know I see many familiar faces, or sorry, names on the attendee list. And so obviously many of you are very familiar with the concept, but every week we get new people. And so just to kind of summarize what we're doing here, this platform trial is testing multiple drugs, or I should say we started testing multiple drugs in 2020, and we're adding drugs as they become available, which is important because we want to test as many drugs as possible in a rapid time frame. And so we started with the first regimen in 2020, and now in 2023 we are about to add regimens 6 and 7. And when we say regimen, we mean a specific drug and the matching placebo. So every regimen, which is color coded here, is basically representing a different drug made by different manufacturer. And in the context of the platform trial, by using a common protocol, we can efficiently test them. And so we, right now, again, so far, we have enrolled people in 5 regimens, and specifically the number 5, or regimen E, which is a regimen testing trialose, which is made by Silos Therapeutics, is really super, super close to completing enrollment. I expect that we will be able to finalize the numbers and make announcements very soon. We are working across a network of 60 sites, and so we depend on sites to enter data and give us feedback as well. So again, I do expect that we, as you know, we've been getting very close to completion of enrollment. And as soon as that's kind of official and we receive all the information from all the sites, we will let you know. But basically, we are at the point essentially of being complete. And so when we can officially, get all the data and announce completion, that basically starts the clock, because we can only get to analyzing results only after all the people who have been enrolled complete the first 6 months of follow-up. So essentially, from the time we complete enrollment, 6 months later, we will have completion of a follow-up, and then it takes a few weeks to get all the data in, clean the data, analyze the data, etc. So that's really exciting that we are making progress towards that goal. The other important event or set of events that's happening, and we're really working hard on those, is to really add new regimens. We always want to have spots available for people with ALS who want to participate in the trial. And so we are now in startup for regimens F and G, which are number 6 and 7 respectively. The manufacturers and industry partners working on these regimens are Calico and Denali, respectively, for regimens F and G. Both are in startup. We expect regimen F to start enrollment sooner, and then followed rapidly by regimen G. So we will have 2 regimens available at any given site. We're operating now on over a large network of sites, and we expect most, if not all all sites to participate in both regimens F and G. So that will open up hundreds of spots for participants. I do want to again share that we are very excited. We're starting to train sites so that they can get activated, and we expect that to happen in sort of the February-March timeframe for F and April-May timeframe for G, which means that basically in just 2 or 3 months, we will have hundreds of spots for people available to enroll in regimens F or G at their institution. Again, it's a large network of sites. We had 60 sites participate in E, but we expect even more sites, around 70 or so, to participate in F or NG, I'm sorry, both of them at all sites. So the reason I'm saying this is because I know some people, obviously whenever we have a gap in enrollment, that means that we cannot enroll new people as we transition from E to F and G. However, again, F and G are becoming available very soon. So if you didn't make it into E, you haven't had a chance to enroll yet, I think, F and G again are coming very, very quickly. So if we can go to the next slide. So how do we test each regimen? And the schema is the same. So essentially, we have first of all, people need to be screened for eligibility. And if they're eligible, they will be assigned to active or placebo within that regimen for a randomized period that lasts 24 weeks or about 6 months. And after that, that's followed by what we used to call open label extension. And for F and G, the real the best nomenclature really is active treatment extension. And why is that important? So I do want to emphasize that the randomized period and the active treatment extension are essentially one trial. They're both incredibly important to gather insights about the drug. So during the randomized placebo-controlled period, participants are randomized in a 3-to-1 ratio to active or placebo, which means that many more people get active compared to placebo. And then that period allows us to evaluate all the endpoints of the trial. And then we give everyone who completes that 6-month period the opportunity to continue to extend the treatment using active drug. And so then we will be in a position to compare people who started on active right away to people who started on active 6 months later. We're basically embedding a delayed start design in the trial, which has been used in many other trials, and specifically also trials that have been successful. So like the Centaur trial, for example, that led to the approval of Relivir. So we're very excited that we can embed that part, give people access to active as part of the active treatment extension, and really comparing the people who started early to the people who started later. Next slide. So the platform trial is also, and that's true for most platform trials that I know of, it's also an opportunity to advance science. And we specifically designed the HEALEY ALS platform trial to test drugs, obviously rapidly and efficiently, but also to advance science. And that's important because there's still a lot that we need to learn about ALS. And so if we use trials as an opportunity to understand ALS better, then we are in a better position to, first of all, to evaluate the drugs that we are evaluating, but also to plan adaptation to the trials that can make it better and plan and help plan other trials and advance the science. And so because of that, everyone who participates candidly donates a lot of samples and data so that we can all learn together as a community. So specifically, we have done a whole genome sequencing on all participants so far, and we are in the midst of analyzing that. We have been measuring neurofilaments for everyone who has been participating so far, and we will continue to do so. And we are now analyzing that. We have a host of biomarkers that are drug specific. And so within each regimen, there's sort of a set of additional biomarkers that can be collected based on that particular drug. We have included 2 digital biomarkers. So digital biomarkers are basically biomarkers that we can measure in the home environment by using a smartphone and some applications. And so essentially we used 2. For the majority of the regimens, they have included speech analysis using an app. That's very important to be able to monitor speech again at home. And so we are analyzing that. And then during the pandemic, it was particularly important to use home spirometry, which is a way to measure respiration at home. And the reason it was important during the pandemic is because many hospitals were restricting basically the use of respiratory devices in the hospital due to the concerns that it could spread COVID. So we are not using it right now because it's no longer needed, but we certainly, first of all, we would have not been able to do the trial during the pandemic without this. And so now that basically we're emerging from the pandemic, we're studying, analyzing all the data to see if it's worth reinstating it, or maybe using home monitoring instead of some of the in-clinic procedures. So more to come on this, but certainly lots of knowledge. All of you have participated in the trial so far, have contributed all of this data in the background that's now we're analyzing. It's an enormous amount of data, and we're planning a number of presentations and publications. You'll hear all about it on these webinars, and we'll be sure to share all the results as soon as they're available. And for the new regimens, we're also planning new outcomes and biomarkers, because new science gets developed and we want to incorporate it. And specifically, some of you may have helped develop the ROTS questionnaire. ROTS is a patient-reported outcome that people can easily fill out at home, and we will be including that for F and G. So that's a way to go beyond, what the ALS-FRSR and other questionnaires do, and really try to incorporate the patient perspective more into the trial. And that was developed by Dr. Fournier at Emory University and colleagues. So thank you very much again for all this donation. Essentially, by participating in the trial, you really contribute to research in a great way that can have a big impact on everyone who has ALS. Next slide. So one question that we've been getting is, how do you select the drugs that go into the platform trial? And so I just want to share again, for those of you who have not been here before, that all drugs that are in the trial are chosen by a committee of expert ALS scientists and clinicians. Well, first of all, a company needs to submit an application to be in the trial. So, we need companies to be interested in participating in order for us to choose them for participation. And so if they submit an application and are then selected, then we work closely with a number of colleagues to tailor their new regimen to their drug. And then again, we add the drug to the trial. So by definition, there's really equal chances of each of these drugs to be positive. So we don't know, again, which drug will be positive. It could be one more. And from our point of view, they're all equivalent, because they are all chosen based on the same criteria. And the criteria are robust preclinical data, which means that all of these drugs have been tested before in the lab, supporting the rationale to go into human testing. And also, all of these drugs have had some set of human experience before, either in ALS or in other neurological diseases. And that's important because, this is a large trial. And so we need to know a lot about dose, safety, and target engagement before we even put the drug into the trial. So those are the criteria. And next slide. So with that in mind, so for in terms of the new drugs that have been selected, so the first one, regimen F, tests a drug that's been developed by Calico in collaboration with AbbVie. That's a drug that targets a regulator of the ISR, or integrated stress response. And there's materials and videos online already available if you want to learn more. We will be sure to have a dedicated webinar on the mechanism of action of this drug with the scientists that actually developed the drug. We've done this for every regimen, for every new regimen. So we invite the scientists to be here and talk about, about their drug, and you can ask questions, and the actual people who did all the science will be on the call. So we haven't fully scheduled yet, and that's because every time we have we share slides with the patient community, they have to be IRB approved, meaning that they have to be approved by the ethics committee that oversees the trial. And so these are obviously complicated slides with a lot of science in it, and so it's taking a little bit longer to have them approved. But as soon as they're approved, we will let you know when the date is, that you can ask questions and you can ask the scientists all the questions about their drug. Same goes for regimen G, the next drug, which is made by, if you can go to the next slide, for regimen G, the next drug is a drug developed by Denali Therapeutics, also targeting the ISR, or the integrated stress response. Again, we have some material already on our website. I know Catherine has kindly posted some information about the press releases, so you can go to those links to learn more. You can use the QR code or the link on the slide here to go to our website. And then again, also for the drug made by Denali, we will have a dedicated mechanism of action, a scientific webinar with the people who developed the drug. So you'll have opportunities to ask questions questions to both Calico and Denali about their specific drug. Next slide. And so also we record all of this. So if you have questions about previous webinars, some are our regular weekly webinars, some are they have a specific title if they were about a specific topic. So for example, you know, based on this example here on January 26, we spoke about regimen C in particular. So if you're interested in that particular regimen, you can always watch that recording. You can also download the slides. At this point, we have, I don't know, hundreds of hours of recordings and slides, and really lots of material for anyone who has interest in ALS research, and in particular in this webinar. And I see that Catherine posted, that's actually a very important link. I would ask you, you know, to sign up for the ALS link, which is our newsletter, because we always post information about webinars. And so if you want to receive information, you know, you can also just go on our website and look it up. But if you want to receive information directly, you can also sign up for the link, and please feel free to share it with your friends as well, or on your social media. Next slide. And as always, please contact Catherine and Alison if you have any questions. I don't know, Catherine or Alison, anything that you would like to say to people so that they can, you know, best communicate with you?
No, Allison is a good presence on social media. So for people who are active on Facebook, you might see Allison around there. I ironically am not much of a social media presence, but I am always available via email or phone. So if you reach out to that email address or the number on the screen there, and I'll put it in the chat, that's me on the other line, and I'm always happy to connect if you have questions about research.
Alison?
I just want to add also, yeah, I'm out on social media pretty frequently because I know that you have questions at any time of the day or night. And so we do try to get back to you as quickly as possible, regardless of what time or day of the week. So please feel free to reach out at any time.
Great. There's a couple of technical questions. I'm going to take them rapidly. So first of all, the Emory tool is called roads, like a street, a road. And actually, I think that Alison posted the scale. So you can go on the Emory University website in the Department of Neurology, and they actually posted the document, the actual PDF, and they also published a few papers. So feel free to use the link to go there and see it. And then there's a great question about the technical issues. So somebody was watching the recording and said that we need to move the speaker boxes because it was cutting off the slides. That's a great point. I'm gonna ask maybe Jesse and Catherine and Alison if we can maybe take a look at some of the recordings, and maybe we can figure out how to place the boxes. I'm not sure how to do it, but I'm sure that there must be a way. So thank you so much for letting us know. In fact, one thing I was thinking the other day, I generally don't watch this as an attendee, so I'll be sure to do that one day, because I want to make sure, you know, maybe we can take turns, because definitely if there's any technical glitch, we want to catch it. So thank you so much for letting us know. There's a great question about, you know, so which other neurological diseases may be sort of close to ALS to get, you know, good data about the drugs that are then tested in ALS. So essentially, you know, I think that's a really important question. So I said earlier that each of the drugs that have been tested and continues to be tested in the platform trial have been tested to some extent either in ALS or in other diseases. And so specifically, you know, there's been a few drugs that have been tested in other neurodegenerative diseases. And that's, I would say the neurodegenerative diseases are probably the closest from this point of view, because the neurodegenerative diseases such as Alzheimer's, for example, or even Huntington or Parkinson's, they do share a few pathophysiologic mechanisms. They're not identical, obviously, because otherwise, you know, it wouldn't make sense, you know, that are different diseases, but they share at least some pathways in common. In fact, for example, Relyvrio, the drug that was just approved for ALS, has also been tested for Alzheimer's. Again, there are some mechanisms that are shared, and so it makes a lot of sense that these drugs are tested in other diseases as well. in fact, there's a kind of follow-up questions about regimens F and G. So they've already been tested actually in small ALS trials. At least the calico drug has been tested or is being tested in a separate ALS trial. And for G, I'll be honest with you, I don't remember right now, but we can definitely look up the information about which other diseases they've been tested in. I know that again, the small trials in ALS for sure, and then in addition to healthy volunteers and a few other diseases. Great. And again, for all these new drugs, we're going to have a lot of information coming up by way of dedicated webinars with all the companies. That's great. So I think those are the questions that I saw this week. And again, always great to see everyone. In fact, today I saw somebody in the clinic who told me I'll see you tonight. So thank you so much for being here today. It's really great that we can have this conversation, both one-on-one, but also as part of this community conversation. So thank you so much. Thank you, everyone. Bye-bye.